Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, my name is Aulia. I'm a medical student at the University of Bengkulu. I'm here to present a medical journal entitled Effect of Temperature Towards RNA Concentration, Quantitative Investigation with Spectrophotometer. This journal is written by Febi Fariska Safira and Novi Sylvia Hardiani. Previously, the Honorable Dr. Wisma Yunita MPD as my lecturer. Okay, here we will discuss 5 points. The first is introduction, aim, method, result, and conclusion. Okay, the first we will discuss the introduction. Ribonucleic acid, RNA, is a thermodynamically unstable molecule. The way RNA samples are preserved is critical to maintain maximum yield and quality. Therefore, it is useful for molecular analysis such as real-time PCR. There are many contradictions and variations regarding the temperature for RNA storage. RNA is essential in the normal functioning in eukaryotes and prokaryotes. Isolation of RNA for molecular studies is a common procedure. The expression of certain genes is determined by measuring the RNA, usually mRNA, expression to molecular assays such as real-time PCR. The advancing knowledge on the molecular basis of methylgenesis has led many scientists to come up with gene therapy as the curative option for cancer. Many factors led to the usability of RNA sample in generating a reproduced result. Thus, the ability to store RNA at ease while maintaining its highest quality and quantity is essential. The statement among various studies and even RNA kit procedure is regards to the ideal temperature for RNA storage has been inconsistent. While many consider -18 degrees Celsius as the gold standard, some deduce that storing in -20 degrees Celsius and 4 degrees Celsius fridge is sufficient. In the Laboratory of Biochemistry and Molecular Biology of Faculty of Medicine Universitas Indonesia, we assume that RNA is best preserved at ultra-low temperatures. Thus, -80 degrees Celsius freezer has been continually used to preserve RNA samples due to the lack of data regarding the reliability of -80 degrees Celsius as the ideal temperature for RNA storage. Okay. The next is aims. The aims of this study is to find the ideal temperature to store RNA among -18 degrees Celsius, -20 degrees Celsius, and 4 degrees Celsius by determining change in RNA concentration over 2 weeks of storage time. Okay, the next is method. This experimental study was conducted at the Laboratory of Biochemistry and Molecular Biology, Faculty of Medicine, Universitas Indonesia. 24 RNA samples were isolated from the tissue of rat liver using the generate total RNA mini kit, according to the protocol provided in the kit. Tissues 25 milligram from the 8 different frozen rat liver storage for plus or minus 1 month in minus 80 degrees Celsius were transferred into 24 of 1.5 milliliter microcentrifuge tubes. Cells of the liver tissues were laced with RB buffer and beta-mercaptoethanol by centrifugation using DNA-C and RNA-C. RNA-C-free micropastel until the solution become homogeneous and transferred through the filter column. Ethanol 70% dilute in RNA-C and DNA-C free DDH2O was added and these sets were filtered through RB column onto collection tube to facilitate RNA binding. DNA contaminant was removed by mixing DNA-C into the reaction buffer, which was loaded onto the center of RB column matrix. Samples were then washed with W1 buffer and wash buffer provided in the kit, purified aqueous RNA was suspended in RNA-C free water. The freshly isolated RNA samples were quantified by spectrophotometry using the varioscan flash multimode samples were vortex for a short duration, plus or minus 10 seconds. RNA sample were loaded onto sample holders with RNA-C free water use dilution factor 1:1 ratio. Absorbance was read at 290 and 280 nanometers. Readings on photometric blank subtraction, along with concentration and purity were collected. Afterwards, 8 samples of purified RNA were kept in 4 degrees Celsius fridge, and another 2 batches of 8 were placed in minus 20 degrees Celsius and minus 80 degrees Celsius freezers, respectively total 24 samples. For the following 2 weeks, absorbance from each group of samples storage in different temperature was read on the Sunday every week. All of the thawing process was done as quickly as possible on ice, and the samples were kept immediately into the relevant storage temperature, minus 18 degrees Celsius, minus 20 degrees Celsius, and 4 degrees Celsius refrigerator to avoid degradation. Okay, all the all the statistical analysis were done using the SPSS software with parametrical enough tests for parametric data and critical Wallis parametrical Wallis test for non-parametric data. Okay, the next is the result. Sorry, but okay, the next is the result. Samples stored in 5 degrees Celsius show a slight fluctuation of plus or minus 2 microgram per milliliter after a week and stayed relatively the same on the second week. Similarly, mean RNA concentration in minus 20 degrees Celsius was plus or minus 3 micrograms per milliliter higher on the first week and decreased around plus or minus microgram per milliliter on weeks 2. The mean concentration of RNA samples kept at minus 80 degrees Celsius rise by plus or minus 3 microgram per milliliter on the first week and increased by plus or minus 11 microgram per milliliter on the weeks after. Shapiro-Wilk test showed that the data did not have normal distribution. Non-parametric Kruskal-Wallis test to analyze the sample and the result indicate that there was no significant difference between the storage temperature and RNA concentration. Okay, the last is conclusion. There are no marked change in the concentration of RNA when stored in 3 different temperatures, minus 18 degrees Celsius, minus 20 degrees Celsius, and 4 degrees Celsius over 2 weeks of storage duration. Therefore, RNA may be stored in any of these temperatures without a reduction in quantity. However, there is no indication if the RNA is usable for molecular analysis. It is recommended that if the length of this study should be expanded to at least on one month for the 3 different temperatures, minus 18 degrees Celsius, minus 20 degrees Celsius, and 40 degrees Celsius, and RNA quality analysis through the use of electrophoresis may be incorporated to check to the integrity of RNA sample. Okay, we have arrived at the end of the presentation. Thank you for your attention, and I hope what I have to say can be useful. Assalamualaikum warahmatullahi wabarakatuh.